Thanks for the very kind introduction. So, good afternoon. I'm Chengxin, a faculty member from the Fredolino Lab at the University of Michigan. And thanks, Clara, for the introduction of CAFA just now so that I don't need to go through the basic ideas. So today I bring to you our protein goal term prediction algorithm, StarFunc, which was ranked 5th in CAFA 5. So similar to almost all top-performing Kappa groups, our Kappa pipeline uses deep learning as shown at the bottom of this slide. Additionally, our method, StarFunc, makes extensive use of sequence and structure templates from blast search, from structure alignment, protein-protein interaction lookup, and PFAM fab matches, and the predicted goal terms from this sequence homolog structure templates, interaction partner, PFAM families, and deep learning are then combined by a random forest model to derive the final consensus prediction. We may not have the most sophisticated deep learning model, but we try to use the template information to the fullest. So in the following slide, I will explain these component methods that use templates one by one. So the first component method is based on sequence homologs detected by blast search, where the prediction score for goal term Q based on sequence is calculated by a weighted k nearest neighbor shown in this equation. Here, the denominator sum over bit k and idk, which are the bit scores and the sequence identity of the k blast hits. And the denominator are the subset, summation of bit scores and identities for the subset of blast hits with the goal term of interest. So we choose this particular scoring function based on our recent large-scale study. So in this recent study involving goal term predictions for thousands of proteins, that low knowledge and limited knowledge proteins, we compare 11 different scoring functions where the first 6 scoring function weights the top k templates based on bit score, different definitions of sequence identity, or combination of bit score and sequence identity. The last 4 scoring function just takes the maximum value of the of the different forms of sequence identity. So for almost any sequence search tool such as BLAST or Diamond or MM6, and for all 3 goal aspects, MF, BP, or CC, scoring function S2, which combines bit score and sequence identity, consistently result in better performance. So the second component of StarFunc is based on structure templates, where the query protein structure is searched by fold-seq through the AlphaFold and BioLib2 database. So here, BioLib2 is a subset of experimental PDB structures with function annotation. The top fold-seq hits from the AlphaFold database and BioLib2 are then realigned by TM align to get the TM score for the K template, TMK. The predicted score for goal term Q is calculated also by weighted k-nearest neighbor, where each template is weighted by bit score, TM score, and sequence identity. Thanks to the introduction of the fold-seq pre-filtering before TM align, the re-implementation of this structure-based pipeline in StarFunc is much faster than our previous structure-based function prediction pipeline cofactor, where cofactor takes around 10 hours per protein, while StarFunc only takes 16 minutes. So this allows us to apply structure template search for very large-scale data, such as those used in Kappa 5, which we previously could not perform in Kappa3 or Kappa4. The third component of StarFunc is based on protein-protein interaction from the string database. So in this pipeline, the query sequence is first mapped to the big string database by blast search, and the protein interaction partner of this blast hit is then looked up through the string database to get the Go annotation annotated to this interaction partner by the string database. And the Go and the Go term prediction is is derived from the top K string interaction partners with the highest string score, which represents the likelihood of their interaction with the query protein. So one problem of using the annotation from the string database is that the string database does not differentiate experimental versus computational Go annotation, such as those with IEA evidence. This is why we implement another pipeline where the Go terms are derived from a smaller subset of string interaction partners that can be mapped to experimentally annotated UniProt proteins. Then the overall prediction score for a particular Go term is a weighted average from the two pipelines that use the small database and the bigger string database. So the fourth component method of StarFunc is based on PFAM families identified by hidden Markov model scan of the query protein through the PFAM database. We train one logistic regression model per Go term, and the feature XK indicates whether the protein sequence matches the K family in the PFAM database. So the last component method is based on deep learning model interlabel Go, which is developed by Chengxin, who will make a more detailed presentation on the model later in this afternoon. So I'm not going into detail on how the deep learning model works here. On our independent test set of 2,475 no knowledge and limited knowledge proteins, the final StarFunc pipeline is consistently better than the preliminary version HFM7ZC used in Kappa5, and which in turn is better than other existing deep learning models shown in gray and template-based methods shown in white. And StarFunc participate in Kappa5 as team HFM7ZC, while the deep learning model interlabel Go participate as team Evans, and the two teams was ranked 5th and 6th around more than 1,600 teams. So here during Kappa, the StarFunc just use an existing deep learning method, SPOF-Go, while it try to experiment on what is the best way on using templates, while the interlabel Go method in Kappa is a full implementation of our latest deep learning model. So the two teams, they are actually does not overlap in terms of methodology. So given the decent overall performance of StarFunc, we wonder which of its components makes the most contribution to the final performance. So individually speaking, the PPI method has the worst performance compared to any of its other 4 component methods, as well as the final consensus StarFunc prediction. However, in an ablation study where we remove one method and see the drop of performance, so we find that the removal of deep learning method unsurprisingly causes the biggest drop in overall performance, followed immediately by the removal of protein-protein interaction. So this suggests that even the PPI method is not great by itself, but it shows a strong complementarity to other components of the StarFunc method. So as a case study on the relation between component methods and final consensus prediction, we focus on a case study on a transporter FHUE from E. coli, shown on the left. So the StarFunc's component method predicts one correct goal term, iron coordination entity transport, as shown in panel B, as well as an incorrect goal term, amide biosynthetic process, shown in panel C. After the consensus prediction by random forest, the StarFunc prediction score for the correct goal term is higher than any of its component methods, while the final prediction for the wrong goal term is lower than the component methods. So this is why for the StarFunc achieves a higher weighted F measure of 0.73 shown on the right, which is better than any of its component methods. So this case study illustrates that the consensus prediction by random forest does not simply aggregate the prediction of all components, but rather try to differentiate correct versus incorrect prediction by component methods. So in summary, our study shows that both deep learning and template are important components of protein function prediction. We also find that template-based method, especially those based on sequence homology, depends heavily on the design of a good scoring function. And finally, our StarFunc does not use expression data or text mining, which, as shown by Clara, could have a big improvement to the final performance, and we aim to add them to future version of StarFunc. So if you are interested in our StarFunc method or interlabel goal, you are welcome to check our web server and the first two preprints. If you are interested in how to design a good scoring function for sequence homology-based function prediction, you can check the last paper that are just accepted by Briefing in Bioinformatics. With that, I would like to thank the other two members of our Kappa5 team, Dr. Fratellino there and Chengxin Liu over there, as well as all members of the Fratellino lab. And Chengxin is going to present later this afternoon, and my other student, Junzhe Guo, is presenting a poster for RNA loop modeling, and we have another Vivian presenting for the evolutionary computational genetics. And thanks for your attendance.
Questions? Thank you for the talk. I have just a curiosity. Why did you use only protein-protein interaction data in string as opposed to other lines of evidence like co-occurrence, neighborhood, so on?
So the question is for the PPI, why do I use the string database in particular?
Why use only this evidence and not the other evidence, the line of evidence that you have in string?
Well, for the
it's not only protein-protein interaction string.
So we in the string we just use for the interaction. We didn't differentiate different evidence. Right? So we use both experimental and computational prediction, including those that are functional association found by genome neighborhood. But we try to differentiate experimental versus computational function annotation for the
So in that case, maybe you couldn't use the protein-protein interaction because it's only a part of the evidence in string.
I do not fully understand your question because string is an interaction database. Maybe we can talk offline.
Okay, we have an online question. How did you handle proteins which are not in the string database, but they are in UniProt?
If they are not in string, then we can use it for the PPI in particular. But since we have multiple, like we have multiple components that hopefully will mitigate that issue. But for the CAFA dataset in particular, almost every target we can find in string because it's mostly Swiss Pro or model organisms. So that's not a big problem for CAFA.
Great.
Thank you for your great talk. I like the idea that span the training data with multiple perspectives, but also in a sense, just a simple question. I'm just wondering, while expanding the training set, is there any case of data leakage for the test set?
Well, we didn't use text mining, so hopefully there's less data leakage. So when we curate our data set here, so we also perform something like CAFA, where we do a time-elapsed experiment where the training set is annotation before 2022, up to 2022, and then the validation is between 2022 to 2023, and then an independent test set is that later. But there could be some sequence similarity between the test and validation, but we didn't differentiate here. But we do have supplementary figure that in the paper that we try to differentiate.
Thank you. I have a small question regarding the scoring functions. When you are using your sequence, your BLAST, how are you dealing with the domains, multi-domain proteins?
You mean how do I?
I mean, is there any alignment coverage thing?
Yes, but not really. So when we calculate, so here in this scoring function, the sequence identity here is a global sequence identity normalized by the longer of the two sequence. So if only a small chunk is completely identical, but it's only a small portion, then the sequence identity will be low. So that's the way we try to consider that.